Welcome, everyone. Thank you for tuning in. I'm Casey Comboy. I'm a second-year student here at Lehigh University, where I'm studying biochemistry, and where I was also part of the CPhages 2022 cohort, as led by Dr. Vassie Ware, Dr. Margaret Kenna, and our graduate student, Stephen Mensa. Today, I'm going to be presenting and sharing with you the work of my cohort, specifically in bioinformatic discoveries, investigating mycobacteriophage genomic relationships and immunity patterns. So my cohort was able to isolate 18 novel bacteriophages using the host M. smegmatis, and we did so following standard isolation protocol from environmental soil samples, as you can see outlined here in this schema in the bottom left. So we isolated from environmental soil samples to obtain pure plaques that we then used to extract DNA and image with electron microscopy. From this, we were able to identify one Mycobacteriophage morphotype, which was phage pescara, seen in image K here at the bottom or top right of the screen, excuse me, and all the rest of the phages were identified to be of the Cyphoviridae morphotype. At the end of our discovery semester, we decided on two phages to be fully sequenced that seemed unique to each other and also to the other phages in the class, and that was my phage Cullens and phage blue crab. All the rest of the phages then had their DNAs pooled for Dodgem sequencing as well. So in the bioinformatics part of our course, our main focus was fully annotating the sequences of phage Cullens and blue crab. And we did so using bioinformatic tools like DNA master, Famirator, and BLAST functions in order to assign functions to each of the features. With this data too, we were able to directly compare Cullens and blue crabs. So for instance, both were hypothesized in the discovery part of our course to be temperate phages. And with our annotation data, we were able to confirm that they were temperate phages since they had an integrase, an immunity repressor, and exonuclease, which are all enzymes necessary to form a lysogen. But being that Cullens is an A3 cluster and blue crab is of the F1 cluster, there are obviously some differences to come from that too, which was also highlighted in our annotations. So for instance, in this middle panel, these Famirator images just go to show some unique features of blue crab versus Cullens. So for instance, in blue crab, a crow protein was actually identified, which is kind of interesting since the crow protein is not necessarily typical of temperate phages, but it was consistent with other phages, F1 phages similar to blue crab like Joey Jr. Something unique for Cullens was also a cluster of 3 tRNAs in the beginning of the sequence, as you can see right here, which was consistent with other A3s as well. In addition to these annotation data specifically for Cullens and blue crab, our class also worked with the Dodgems data. And what we did here was we decided to focus on one fully sequenced contig that was identified as a cluster K2 using BLAST and designed primers specific for this to run a PCR. So that's exactly what we did. We ran a PCR with all the phages lysates with the K2 specific primer. And what we found was that only one phage from our class, seen in our gel right here to the right of my screen, yielded the 578 base pair band that we expected to see in lane 7. And this was phage donut. So using Dodgems, we were able to identify another phage and their cluster. Another part, while we were doing all this annotation, we worked to isolate or make lysogens from as many temperate phages as we could. And the way we did this was we isolated lysogens from purified bacterial colonies and formed a putative temperate phage, which we then tested with their abilities to form halos. We did those halo tests with 3 rounds of purification before testing its ability to release phage in a supernatant when grown in liquid culture. And with these lysogens, then we were able to form lawns. And with these lysogen lawns, we were able to compare phages on a class-wide scale with what's called an immunity assay, where we took the phages and serial diluted them and plated them on these lysogen lawns. And the results of this assay is seen here in the middle bottom panel, where we have this table of all the EOPs. And the most interesting point, one of the interesting points from this assay that I want to talk about today is the relationships between colons and frumpkins. So if you come here to the future directions panel, you can see that the colon's lysate was not able to form, or I should say, not able to infect a frumpkin lysogen lawn, and nor was frumpkin able to infect a colon's lysogen lawn. So this homoimmunity led Cal Schutak, who discovered and isolated frumpkin, to believe that frumpkin could also be an A3 cluster. So he did some deep diving back into the DOGEMS data, and using BLAST was able to narrow down his hypothesis to context 2 or 6 being one of the sequences that belonged to frumpkin. And this is something that we would want to further investigate, possibly by designing primers specific for these sequences or specific for frumpkin A3 to run a PCR with frumpkin lysate to be able to say for sure whether or not frumpkin is an A3 cluster phage and to be able to correctly identify which contig belongs to frumpkin. So that's all I have for today. Thank you for tuning in again, and I hope you were able to learn something.